Hello students and welcome to one more lecture of genetics. First of all, I want to say congratulations for getting towards the end of this course. This is our very last lecture. This is probably the last time you will listen to me through a screen of a computer, okay? And to make this class a little bit more interesting, because I do know that towards the end of this short semester we are probably a little bit tired, I decided to talk a little bit about some exceptional topics in genetics, and one of them is about cancer. So we are gonna talk about the genetics involved with cancer and also some advances in genetics, and we are going to talk about some genetic tools that we use in research, for instance. Okay, so I hope that you find this class more interesting than the previous ones, because I know that the previous ones were full of concepts and sometimes they can be a little bit tiring, but this one you will notice that it's a little bit lighter. Okay, so I want to start this class by talking about the genetics involved with cancer. Cancer is the second common cause of death in Western countries. We can understand cancer as a genetic disease at a somatic level. What does that mean? What is a genetic disease at somatic level? That means that the genetic disorders involved with cancer will happen with your somatic cells. Most of them will happen with somatic cells. Okay? There are some cancers that happen with the germline cells, but they are less common. Most of the cancers that we have are attacking specifically somatic cells, or cells that are diploid. Okay, you remember those concepts. And they are characterized by gene products derived from mutated or abnormally expressed genes. So by mutation, we've talked about some forms of mutation in last class, right? When we have, for instance, a replacement of a base pair in a sequence of the gene, we can have the final product of that gene mutated or changed, right? So the final product is the protein. Therefore, if there is a mutation related to that gene, we can expect some differences in the protein, in the product that come from that gene. Some of these cancers can be inherited, but most of them are created within somatic cells. And these somatic cells will divide and form tumors. Okay, and we are going to talk a little bit more about tumors in the next few slides. Most of the therapies that we have today to treat cancer are chemotherapies to actually cause the death of those cells. However, there is a certain expectation that in the future we'll be starting to use more gene target therapies to actually defeat those cancers, because we do know now that most of these cancers are actually happening due to gene abnormalities, okay? Mutations happening at the gene level. All those factors, for instance, smoke, right, will cause some sort of alteration at the DNA level. So we are now trying to advance our current treatments to target those alterations at the DNA level, okay, which can be more beneficial because, as you might be aware, chemotherapies can be very dangerous. Not dangerous, but can be very harmful to the patient, because it ended up killing not only bad cells like cancer cells, but also sometimes some good cells. And that's why there are so many side effects. So here we want to understand cancer as a genetic disease that happen at the or that happen with somatic cells. Some of them will be associated with the germline cells. Most of these cancers that we have knowledge about are associated with somatic cells. The genomic alterations that can be associated with cancer cells can be, for instance, single nucleotide substitution. That's a type of mutation that we studied in the previous class. For instance, remember, you have your DNA strand, and then in that DNA strand, one nucleotide is replaced. That can change the entire gene product coming from this triplet codon. Remember this concept from last class? Which can then change the amino acid associated with this triplet codon, which can then change the protein. There are some genetic alterations also associated with the cancer cells that happen in a large-scale chromosomal rearrangement, literally meaning that the chromosome that we know with 2 arms can have some alteration in a certain portion of this arm of the sister chromatid, and that can alterate the entire configuration of the chromosome. Some of the genetic alterations associated with cancer cells are also due to amplification and deletions of genetic sequences. Most of the cancers are caused by mutations associated or present in somatic cells, but there are some types of cancers also attacking or happening with germline cells, okay? Germline cells are those cells that lead to the production of a new individual, right? So here we have the eggs and the oocytes. Sorry, the eggs and the sperm. Oocytes and eggs are the same thing. Okay? There are 2 fundamental properties of these cancer cells that make them bad for the human body. One of them is proliferation. Proliferation refers to the abnormal cell growth and division. It's when one single cell grows abnormally, and not only that, that cell also starts dividing in multiple cells due to mitosis. So there is no control here of the mitotic process, okay? Mitosis happens in an abnormal way. The second fundamental property of the cancer cells is metastasis, and you probably have heard of this. So this happens when the cancer cells starts spreading and colonizing other parts of the body. And that happens because the machinery of the cell that keeps the cells together starts failing. So one cell is attached to the other cell due to adherent molecules, okay, that keeps them together. However, if this is a cancerous cell, maybe the machinery that keep the cells together and attached to each other starts failing, and therefore this cell now is able to spread to another part of the body where it becomes where it colonizes, okay? So metastasis happens when the machinery that keeps the cells together restrained are not functional anymore. Therefore, they detach from their original tissue and spread and colonize other parts of the bodies, okay? Another important concept that probably you have heard is the difference between the benign and malignant types of tumors, okay? Benign tumors are those types of cancers where cells lose their genetic control over their growth. So they grow abnormally, okay? and that will result in a multicellular mass. So you can think of one cell in that tissue that, for instance, starts growing and also dividing without any control, okay? Which results in a multicellular mass in that tissue. However, this cancer is not very harmful to the body and therefore can be easily removed with surgery. So we can mechanically, physically remove these cells from the adjacent tissue, okay? And therefore protecting the remainder tissue that stays in the area. However, the malignant cells, or the malignant tumors, happens when those cells are no longer able to be kept restrained in that region, and they detach, and they ended up spreading and colonizing other tissues. And therefore they form what we call secondary tumors or secondary metastases, okay? And when they start invading other tissues, when they start colonizing other parts of the body, then they become more life-threatening. There are benign tumors that can actually advance to a malignant tumor, but most of the malignant tumors that we observe are those that actually spread throughout the body and colonize other areas, affecting other areas. Also, another concept that is important to understand is that all cancer cells in primary or secondary tumors are clonal. What does that mean, being a clonal cell? That means that those cells originated from a common ancestral cell that accumulated different various mutations. So one original cell in the body starts suffering lots of mutations, okay? Single nucleotide replacement or deletion, rearrangement of gene sequences. Many various mutations start happening with these cells, with the ancestral cell. And then that cell, when it goes under mitosis now, produces two clonal cells that accumulated all these mutations. Those other cells, as you can see, will undergo a new mitotic division and will also carry the mutations that the original cell developed. So all these new cells are known as clonal cells, and they came from the original cell that accumulated specific mutations. All right? But more than just accumulating mutations, we can also understand that the cancer is a multi-step process. So that means that to be a cancer or to be life-threatening to the body, the cell requires multiple mutations and multiple clonal expansions, okay? Because one single mutation or a point mutation in the genetic sequence of the cell is not enough to actually cause a tumorigenic malignant cell, to cause a cell that is actually gonna become a malignant tumor, a tumor that will be life-threatening. Because like we've studied in the previous class, most of our cells have a specific machinery to take care of those mutations, right? Our cells, they have specific DNA repair tools that will, like the name suggests, repair the changes in the DNA sequence, the changes in the genes, okay? Which can then also prevent changes with the final product of that gene. So point mutations or single mutations sometimes will not lead to a malignant cancerous cell. We need actually several mutagenic events in that one single cell. Okay, so many mutations happening in the DNA sequence will actually lead to the development of a malignant cell. All right, because single mutations can be easily repaired by the cell machinery. Okay, so many mutations in those cells are necessary to actually cause a malignant tumor or even a benignant tumor. All right. Cancer cells also contain genetic defects, and those genetic defects, they affect the genomic stability. Oops, I guess my pen did not work. They affect the genomic stability of the DNA. By genomic stability, we can understand as a normal composition of the genome. Okay, so let's say that the sequence in your DNA in your strand of DNA is AATG. Maintaining this sequence refers to genomic stability. Okay, some of the cancer cells also have means to bypass the DNA repair mechanism. So those mechanisms that we studied in the last lecture that helps repairing the defects that happens to the genes, they can be bypassed by cancer cells. Some of those cancer cells will lead to will also present some chromatin modifications or modifications in the general structure of the DNA that leads to changes in the whole chromosome formation. Okay? Because of that, we have a mutator phenotype. What is that mutator phenotype? A mutator phenotype is a certain type of cell that shows a high level of genomic instability. Usually, those cells are cancer cells. So cells that have high levels of genomic instability, they can lead to the development of cancer. And that's because the genome in those cells are no longer stable. So cancer cells, they do have a higher than normal rates of mutation and also chromosome abnormalities or instabilities. So mutation, we can think of all the examples that we already studied in the previous class. In chromosomal abnormalities, we can think of the general structure of the chromosome changing. These two factors lead to changes as well in the genomic integrity of that DNA. Changes in the genomic integrity of the DNA increase the rate of mutation for that specific DNA. And those cells that have increased rate of mutation, we call them, or we say that they have this mutator phenotype. Okay? This genomic instability in those cancer cells or in this mutator phenotype is characterized, like I said, by mutations, right? And those mutations are usually somatic point mutations, or also, like I said, chromosomal abnormalities. Those chromosomal abnormalities can be translocation, aneuploidy, chromosome loss, DNA amplification, and deletions. All these factors can change the structure of the chromosome. Okay? Another important factor that we must remember about cancer cells is that they bypass the normal machinery, the normal tools that the cell uses to regulate the cell cycle. By cell cycle, we're talking about mitosis and meiosis. Okay? And we've studied, I believe, in our very first class, that the cell uses specific molecules, for instance, the cyclines and the cyclin dependence kinase. I think those were the two ones that we talked about that help regulating the cell cycle, right? The transition from one phase to the other. However, cancer cells have means to bypass those molecules. Therefore, the cell cycle is no longer regulated, which leads to this cell proliferation or abnormal cell proliferation, okay? Because normally the cells proliferating or the cells increasing in number, duplicating, it's a okay process. It's a process that we need in the body for repair of tissues, for instance, right? When you have a wound, those cells in that region, they must multiply to remake, to redo that tissue that you lost. The problem is that the cancer cells, they lose the control over this cell proliferation machinery. They lose the control of the molecules that help them regulate their cell cycle. So instead of just, healing that wound, the cells would proliferate abnormally, and then that would cause a mass of cells in that region, okay, leading to the development of a tumor. Okay, there are some cells that they do continue to grow throughout the lifetime of organism. However, it is important that the cells keep this cell cycle regulation machinery under control, just so the cell knows when to start the G phase, when to finish the S phase, and when to transition to the mitotic or meiosis phases. Okay? So, remember, in the cell cycle, we have 3 checkpoints. Those 3 checkpoints are points in the cell cycle where the cell monitors external signals and the internal equilibrium. By monitoring what's happening outside and what's happening inside of the cell, they are able to decide whether they will proceed to the next stage of the cell cycle. What are those checkpoints? The first checkpoint that we have is between the phase G1 and S. The second that we have is between the G2 and the mitotic or meiotic phases. And the last is during the mitosis or during the meiosis. Okay, so if we represent the cell cycle like this, the first phase is the G phase, G1 phase, then we have the S phase, then we have the G2 phase and the M phase. M phase here can be mitosis or meiosis, okay? The first checkpoint will be between G1 and S phase to make sure that the cell has normal internal environment. Then the second point will be between G2 and the M phase, and the other checkpoint will be within the M phase. So the first checkpoint that we have, which is between the G1 and S phase, is to monitor the cell size and determine whether the DNA suffered any damage. Then the second checkpoint that we have is in between the G2 phase and the M phase. And in this checkpoint, the cell will check for the physiological conditions. For instance, what is the nutritional state of that cell? Does that cell have enough food, right, so to speak? Does the cell have enough food that it can continue to the next stage and it won't starve, for instance? Okay? Then the other and last checkpoint is during the M phase, and here the cell will check for the formation of the spindle fibers, and those spindle fibers help attaching to the kinetochores. The kinetochores are within the centromeres of the chromosomes, okay? And those centromeres of the chromosomes are also monitored. Because, why is that important? Because, let me represent here what I am talking about. Because we want to make sure that when these sister chromatids are pulled apart, they will actually be divided into two different cells. Okay? We don't want these sister chromatids migrating to the same cell, right? We don't want the sister chromatid during the division to be together, because that means the spindle fibers in the kinetochore had issues, all right? And the cell could not identify those issues, which then leads to a mutation in that cell. If you want to know more about the mechanisms involved with the cell cycle regulation, you can watch this video that will guide you through the phases and the processes associated with the cell cycle regulation. Okay? There are two specific molecules associated with the cell cycle regulation. They are, like I said, the cyclins and the cyclins-dependent kinases. So the cyclins, the level of cyclins fluctuates throughout the cell cycle, okay? And they lead to the activation of these cyclin-dependent kinases. The cyclin-dependent kinases, along with the cyclins, they make up a complex that phosphorylates and also activates proteins. And these processes of phosphorylation and activation of proteins, they will lead to the transition of one phase to the other. So these phosphorylation and activation processes, they are essential for the cell to understand that it has to continue throughout the cell cycle, okay? However, sometimes cancer cells will also have issues in this tool, in this signaling machinery to actually control when the cell has to move from one phase to the other, okay? Which can lead to abnormalities in the cell cycle, which can lead then to, for instance, abnormal proliferation of those cells, okay? Normally, if there is a mistake within that cells, if there is any type of error in the cell, whatever that error means, and the cell can no longer progress through the cell cycle, then what will happen is apoptosis. So naturally, when the cell is too damaged, there's so many damage to that cell, either with the controlling of the cell cycle or controlling the DNA repair. It doesn't matter. If there's so much distress, or if those damages are too severe to be repaired by the cell, then the cell enters in this programmed cell death that we call apoptosis, okay? Apoptosis is a genetically controlled process where the cell will literally commit suicide. So the cell itself has the decision of looking at the DNA and looking at the chromosomal structure and decide whether that is good or bad to continue throughout the cycle, the cell cycle. Okay? Therefore, if these changes are too severe and the cell cannot repair, then apoptosis will happen. That is important because this programmed cell death helps preventing cancer. But as you can imagine, cancer cells, they have ways of bypassing these programmed cell death. Okay? Therefore, the cell that was supposed to die no longer commits suicide. Okay? Therefore, they continue proliferating abnormally. What are the steps involved in this apoptosis, in this programmed cell death? So the first one is the nuclear DNA becomes fragmented. So the cell automatically detects that there is some severe alteration to the DNA. What happens now is that the cell starts breaking that DNA just so that DNA can no longer be duplicated, which makes sense, right? We don't want that DNA to be passed to another cell. Then the second step is the disruption of internal cellular structures. So that means that organelles will start being destroyed, okay? Because again, we don't want to pass those mutated organelles, for instance, to the next cell. Then we have the formation of what we call apoptotic bodies, okay? Which means that the cell now dissolutes or disintegrates into small spherical bodies, small and smaller spherical apoptotic bodies, okay, which are reminders or remainings of the cells. Those apoptotic bodies, then they are engulfed, then they are eaten, let's put this way, by phagocytic cells. So those phagocytic cells will find the apoptotic bodies and they will eat that apoptotic body. And then by eating that apoptotic body, that will destroy the apoptotic body. Okay? There are some proteases that are responsible for coordinating or controlling this apoptotic process. Okay? Those proteases are known as caspases. So caspases are the enzymes that will, for instance, destroy the DNA and digest the intracellular components of the cell. But as you can imagine, cancer cells, they also have means to bypass this apoptotic event. Therefore, the cell does not die, and then it proliferates, and then it leads to the generation of new cells with those unrepaired mutations. Continuing our study of cancer and tumors, we've emphasized already how it is important that the cells, they are maintained or they are kept in their original location, in their original tissue. And then it becomes problematic when those cells, that mass of cells that is growing abnormally, detaches from that original tissue and migrates to neighboring tissues or to other locations. That's when those cells go through this metastasis process. Okay, so a metastasis process is when the cancer cells, they disengage, they detach from their original tumor site. And when they do that, when they leave their original thermal site, they ended up invading other tissues. And when they invade other tissues, they grow and then they cause harm to the organism in that new region. Before the tissue to disengage, to disattach from its original location, for those cells to be to colonize other areas, those cells, they must digest the components of the extracellular matrix and the basal lamina. So if you think of a cell, the cell is surrounded of innumerous proteins, and here I'm just going to represent with another color. Innumerous proteins, and those proteins make up what we call the extracellular matrix and the basal lamina. The interaction of the extracellular matrix and the basal lamina of adjacent cells keep those cells together in their original location and make up the whole tissue. Okay? So here I'm just representing two cells that are connected by their extracellular matrix, and then they are assembled in the whole tissue. The metastasis process happens when these components of the extracellular matrix, which is mainly glycoproteins, they are digested. Therefore, these cells, they detach from their original tissue and they ended up migrating to other regions. Okay? So those tumors that are metastatic, they ended up invading other tissues by traveling through the blood or the lymphatic system. Okay? So those cells must first digest those components that keep them together. And those components can be different components. For instance, one of them that we have are the cadherin molecules. The cadherin molecules are what we know as a calcium-dependent adhesion proteins. So they keep those cells together. They play a very important role in maintaining this cell-to-cell adhesion, which therefore contributes to ensuring this proper tissue structure. Okay? And that is very important, not only during the development of the normal tissue, but also in adult organisms to keep those cells together. Okay, so these cadherins, they bind to the same type of cadherin on their neighboring cells. So if you think of those two cells that are together in the same tissue, so at the bottom here I'm representing the nucleus. If you think of those two cells, each cell will have these molecules in their basolateral membrane, and those molecules will keep them attached to each other. Some of those molecules are also connected to the cytoskeleton of the cells. Okay, so that all keeps the integrity of the tissue. All right, so some of these adherence junctions that we call this diagram that I just showed you, they are linked to these cytoskeletons to maintain not only like these cells together, but also the tissue integrity and some sort of mechanical stability of those cells. Okay, so cadherins are important molecules that keep the cells together. Here in my example, I'm telling you about the E-cadherin. The E-cadherin is important for the epithelial tissue development. So in epithelial tissue, we find this type of E-cadherin. In the nervous system, for instance, we find the N-cadherin. All right. So in terms of epithelial tissue, a downregulation or loss of this EAT-cadherin can be associated with metastasis of those cancer cells. All right. For the N-cadherin, for instance, downregulation or loss of the N-cadherin, we can actually have wrong neuronal migration or neuronal migration defects. We can also have defects in synaptic function of those neurons. So these cells are very important to maintain, these molecules are very important to maintain cell to cell together. Okay, so if you have issues with these cell adhesion molecules, you can lead to uncontrolled migration of those cancer cells. Not only that, cancer cells can also bypass or can also cause issues with these proteolytic enzymes. So proteo means protein, lytic means digestion. So these enzymes are enzymes that digest proteins, specifically glycoproteins found in the extracellular membrane or in the extracellular matrix of the cells. So when you digest those proteins that make up the glycocalyx of the extracellular matrix, then you are able to make those cells disengage or leave the original mass of tissue. Okay, so these are the main information about cancer and tumors. I know that there are tons and tons of information out there that we could go through, but for the purposes of this class, I think that we covered the main knowledge that we have about cancer. But like I said, you can definitely find more specific information about this topic, especially because each type of cancer will lead to a downregulation or loss or a bypass of a certain mechanism. Here we are talking about generals, okay? So when we talked about the cadherin or when we talked about the other mechanisms involved with cancer cells, we're talking about general terms. But keep in mind that there are many more nuances to how cancers are regulated, to how cancers happen in the human body. Okay? For the second part of our class, I want to talk a little bit about bioinformatics and specifically about genome sequencing or DNA or sequencing of the DNA. Okay, so this is a big picture that we are gonna study from now on. So here you can see those nucleotides kind of paired with other nucleotides, right? So here you have one sequence and you're pairing that sequence with a non-sequence or a sequence of other organisms. So we now, in this portion of the class, we try to understand what is the importance of this sequencing, of this alignment of the nucleotides in understanding the composition of the genome of the organisms, specifically the humans, for humans, but the same principle can be applied to many other organisms. All right? So when we think about the development of the area of genomics, we've got to think that we went from calling those genes as unit factors because we did not know what they were, right? Remember when we talked about Mendel's hypothesis and theories to explain how features are inherited from one generation to the other? He only called those genes that today we know that are genes unit factors. So we went from not knowing what those unit factors were to actually being able to identify each nucleotide that make up the sequence of those unit factors. So when we look at the history and the development of genetics as a whole area of knowledge, we went from not knowing exactly what was happening in the cell and how that information was passed to now we know the precise letters, the precise nucleotides that make up each sequence of genes in a genome. Okay, so we definitely advanced a lot of our knowledge in terms of knowing what's going on with those genes. Okay, and because of that, we branched into too many areas associated with the study of the genome in general. Okay, so first we have the genomics. The genomics is the area of science that is dedicated to actually study the DNA and the gene and the gene, and understanding what are the letters, what are the nucleotides that make up for the sequence of DNA, and what are the, for instance, mutations that can happen to those gene sequences. Okay, then we also have the transcriptome. If you follow the central dogma, transcriptome is the area of this genetics that will study the transcripts, and the transcripts here are the mRNAs. Remember that the DNA is transcribed into RNA, and the RNA is then translated into proteins. So the area that studies the RNA and all the functions associated with the RNA, whether the RNA is expressed or not, whether the RNA will become a protein or not, that is known as transcriptome. Then we also have, for instance, the proteomics. And as you can associate with the name, the proteomics is that area of study that will dedicate to understand whether those RNAs are becoming proteins or not, the functions of those proteins, the structure of those proteins, literally a protein profiling of the organism, understanding what is the role played by those proteins that came from the RNA. Then the other area is also known as metabolomics, because now not only we are interested in understanding the profile of the protein, the structure of the protein, but the function of the protein in the entire metabolism. So this is what I'm telling you. From not knowing what those genes were that contributed to the inheritance of those features that Mendel was studying, now we have different areas in the genetics or molecular biology dedicated to study each step from the simple DNA all the way to the final product of that gene sequence. Okay, so today we have this huge area known as genomic analysis that will allows us to study the genomes of the organisms, understanding not only the sequence of those genes, but also what are the roles executed by those genes, what are the proteins that come from those genes. And with the advancement of this area of the analysis of the entire genome of the organism, we also developed this new area known as bioinformatics. As you can appreciate, the DNA compiles a lot of information, right? So there's a lot of data that can be stored in those sequences. So we need also a lot of a big capacity, we need a lot of ways of analyzing that data stored in that DNA. And with that, we created this, it developed this new area known as bioinformatics, which utilizes mathematical applications to understand the general structure of those genes, to understand the expression of those genes, to understand the structure of those proteins. Okay? I will say that there is a lot that we can talk about bioinformatics and also genomic analysis, but I will try to keep this class as simple as possible, or the final steps of this class as simple as possible, because we are not actually an advanced biomolecular class. We're just trying to understand the principles that led us to understand the sequence of the genes. Okay, so here we're gonna talk about how those genes can be sequenced. How do we know that in that let me get the pen. In that DNA strand, I have A, T, C, G, A, T. How do I know this sequence in that genome? That's what we are interested in. And of course, as you can imagine, this can go much deeper than what I'm gonna cover in this class, but let's just try to understand the general principles of the genome sequencing. Okay, one of the first techniques that was developed to understand the sequence of the genomes was developed by this guy named Sanger. So Sanger developed this sequencing that's known as chain termination method. Okay, so in this first part, I'm going to talk about what is this Sanger sequencing method. Okay, the Sanger sequencing method is also known as D-deoxy sequencing, or known as chain termination method. Okay, so here this method allows us to identify the order of the nucleotides in the DNA. Like I said, we are interested in understanding what are the nucleotide bases that make up for the DNA. This methodology developed by Sanger was actually, or relies on the chain termination by modified nucleotides. So if you think of the chain, and by chain here I'm saying I am referring to the strand of DNA, right? That chain has many letters, okay? And we are interested in understanding what those letters are, what those nucleotides are. Here, we're gonna use modified nucleotides that will stop at known points of this chain the reaction, the chain reaction performed by the PCR. Okay, so here we're gonna use these nucleotides called D-deoxynucleotide triphosphates, and we can simply just call them DDNTP, D-deoxynucleotide triphosphate. These nucleotides, they stop the chain, they stop the sequence of that DNA strand. Okay, so this was the very first DNA sequencing method, and it's actually still used sometimes as a method to certify kind of the new genome sequencing methods that we have. Okay, so over the years, the sequencing technologies, they have evolved, but despite these advancements in the genome sequencing technologies, this method developed by Sanger still is very used, especially in small scale projects, okay? Because it still conserves a lot of precision and reliability, okay, which makes this method very, very, a very valuable tool, okay. We do have these next generation sequencing technology, and I'm going to talk specifically about the Illumina, and you probably have heard about this if you are into this molecular biology area. But you know, even though these technologies that they can sequence large amount of DNAs much more quickly and very cost effectively, we still use the Sanger sequencing for small scale projects and to kind of validate these next generation sequencing. Okay, so this technology, okay, let's talk about the technology itself. So because I told you that it uses this ddNTPs that stop the chain reaction, right? But what does that actually mean? Let's talk a little bit about the technology. So this technique actually involves the incorporation of these ddNTPs into this growing DNA chain during replication. Okay, so we are using a DNA strand and we are replicating this DNA strand. By replication means that we are incorporating, we are manufacturing a new DNA strand, okay? And every time we are adding a new nucleotide, we are elongating this DNA chain, okay? So we are using these ddNTPs that are being incorporated in this growing DNA chain. But every time that you insert a ddNTP into this growing DNA chain, you stop replication. So let me clear the ink here to give you a general idea. Let's say that you have this strand of DNA, okay? You know, you are incorporating A, T, A, but then you now incorporate a, let's see, a C nucleotide base here. And that C actually carries, or that C is this ddNTP, is this modified nucleotide. When you incorporate this modified nucleotide in this sequence that was growing, that I was replicating, then replication stops. And replication stops because the ddNTPs, they lack the 3-line hydroxyl group. And I'm gonna show you the picture of these chemical structures, okay? But by lacking this hydroxyl group in the 3-line carbon, we prevent the addition of further nucleotides in this sequence. So I kind of like stopped my sequence there, okay? So I know that at some point in this long sequence of DNA, I have a C base. And then I do the same thing. So A, T, C, and then I added a T here that carries this ddNTP. So now I know that in that sequence, I have a T nucleotide base. Okay, so every time I add the ddNTP, I stop this sequence. And I know that the last letter of that sequence was C. And I do that, and as you can imagine, we can incorporate these ddNTPs at any point of our DNA sequence. These ddNTPs, they actually carry a fluorescent or a radioactive labeling in their molecule. And by fluorescent, because they are fluorescent, they can be detected. So when I shine a light onto this ddNTPs, I can detect a certain color, and that color indicates that the sequence has a C. This color of the T indicates the sequence has a T. So with that, you can imagine, as you are looking in this figure right here, in the whole sequence of DNA, I can stop that sequence at different points by implementing those DDNTPs. Okay, and each DDNTP here will carry a certain color that I can detect afterwards. And by detecting the color of each of these letters that was incorporated into that sequence, I can then come up with the entire sequence of DNA. Okay, we're going to talk a little bit more about the step-by-step of this process, but this is just a general overview. This method developed by Sanger was very crucial in this project, in this Human Genome Project that you probably have heard, which was a huge effort among the scientific community to identify all the letters, let's put this way, all the nucleotide bases in the DNA of humans, okay? And this was actually completed in 2000, I think it was 2003, probably. In the years of 2003, we had the whole human genome sequenced, and that was actually a very significant milestone in the genetics, right? In the advancement of this area. So like I said, we went from not knowing what those unit factors were to actually being able to completely identify every single letter that's in the DNA, or at least in those genes that are coded into a protein. Okay, so this method gave, a foundation, was the foundation for developing other methods of sequencing the DNA that we now today call next generation sequencing. Like I said, the Sanger sequencing uses these termination of the DNA strand elongation. Okay, so you have a whole sequence of DNA that you want to know the composition. So every time you replicate that DNA sequence, you stop that DNA sequence at a certain letter. Okay, so you have this huge DNA sequence. Once you replicate that DNA sequence one time, you stop at letter T. Then you replicate that sequence again, and now you stopped at letter C. Then you replicate it again and stopped in letter A. Then you replicate it again and you stopped in letter G. So every time you replicate this sequence, you insert a dideoxynucleotide in the sequence, and then you stop the replication. Therefore, you know that in these positions, you have A, T, C, G, and as you can imagine, you can completely know what are the other letters here in this sequence of DNA. Okay? So the principle here is using this, like I said, DDNTP, which stands for dideoxynucleotide triphosphate. These DNTP, they are modified versions of the normal DNA nucleotide. So here you have a normal DNA nucleotide. Remember, you have the A, T, C, and G. Okay? Normally, in their carbon 3 line, they have the hydroxyl group. However, in our modified DDNTP, the hydroxyl group lacks oxygen, which prevents bonding, which prevents forming the phosphodiester bond that would connect another nucleotide here, okay? So let's say that after C, you would connect another nucleotide. However, you can't because now our D-deoxide nucleotide misses the oxygen, and because of that you can't form the phosphodiester bond. Okay, so because of that the DNA strand can no longer be elongated, right? So here in this sequencing process you have a mix of labeled ddNTP. So these deoxide nucleotides, they are labeled with fluorophores, and you can detect those fluorophores in your in certain equipments. The light emitted by these fluorophores can be detected. Okay. Besides of using these ddNTPs, as you can imagine, you also use regular DNTPs, regular deoxyribonucleotides. Okay, so all these other nucleotides that were inserted here are normal deoxynucleotides, normal DNTPs. We also have to use here the PCR, the polymerase chain reaction, because that's what we use to amplify the DNA template. Okay, so the DNA polymerase is an enzyme, and this enzyme will add DNTPs, normal DNTPs, into this growing DNA strand, and occasionally the DNA polymerase will add this ddNTP instead of adding the normal DNTP. And when that happens, the chain elongation reaction stops because you can't form the phosphodiester bond. Okay? So let's look at the whole step involved in this Sanger sequencing. Okay? So first you need to prepare the sample of DNA. You need to collect the sample of DNA. For that, you will need some reagents. For the whole Sanger sequencing to work, you need some reagents. You need your template DNA, which is the DNA that you want to sequence. You need the normal DNTPs, which are these guys right there. You also need the ddNTPs, which are the modified version right there. And as you can see, I am indicating them with this fluorescent light. Okay, so each one will carry a different color. You also need your DNA polymerase and the primers to start the replication. Okay, so the first thing that you need is to collect the DNA. Okay, and there are many ways that you can extract DNA from your from the sample, from the cell. This DNA is then denaturated into single strands. Remember, the DNA in the cell is always double strands, but here you're gonna use only one strand. Okay, so you denaturate the DNA to obtain single strands. Then you go through a PCR, through a polymerase chain reaction. So here, the denaturated DNA is subjected to PCR. During the PCR, the DNA will be mixed with a combination of normal nucleotides and also these ddNTPs. Each of these ddNTPs are labeled with a distinct fluorescent marker. Okay, so for instance, the A is labeled with green, the T is labeled with red, the C is labeled with blue, and the G is labeled with gray. Okay, so the target DNA here is then amplified by using the PCR, And the PCR is a process that includes a initial denaturation followed by cycles of denaturation, annealing, and extension. Okay? We're not gonna go over every one of these processes, but just so you know, denaturation, annealing, and extension are steps of the PCR. Okay? So the PCR mix actually contains the DNA template, primers, and all four of our dNTPs and the DNA polymerase. But we also add in that PCR mix these ddNTPs, and they are labeled with distinct fluorescent markers. Okay? Like I said, the ddNTPs, they lack the oxygen in the hydroxyl group, and then that causes the chain termination when they are incorporated. Okay? Due to their lower concentration in the PCR mix compared to the dNTPs, the chain termination reaction actually can stop at various points, which will produce a collection of DNA strands of different lengths, each one of these DNA strands ending at a different ddNTPs. So this is what I am showing you here. Here are different DNA strands with different lengths because the reaction terminated at various points. So I have a whole collection, okay, of these DNA strands. Then what we have now is separating this mixture of DNA fragments by capillary electrophoresis, because we now want to separate the DNA fragment by size. Okay? Generally, the principle is that shorter fragments will travel faster through the capillary tube than the longer fragments will move more slowly. So as you can see here, I have a tubing where I am putting my DNA sample into that, and the DNA sample by capillarity will be separated in shorter and longer fragments. Okay? As they move through this capillary tubing, we are shooting a laser through the tubing. Okay, so as the fragments pass through this tubing, you have a laser that excites the fluorophore, the fluorescent labels on those ddNTPs. And as you excite them, there is a detector that identifies the fluorescent signal of each ddNTP. Okay, so every time the laser shoots onto the sample, your detector will identify the color, and that will give you this, I think it's called a chromatogram. Okay, that will give you this graph of colors. That's an easier way of putting this. So the sequence of data that you are collecting is then compiled into this chromatogram, okay, which is a graphical representation of colors. Let's put it this way. So this graphical representation shows you the order of the bases in that DNA fragment, okay? The shorter ones are coming first, and the longer ones are the last in that sequence, okay? Because that's how they passed through the tubings, all right? So each peak in this graphical representation corresponds to a specific nucleotide. And then this sequence is then we can read the sequence from the shortest to the longest fragment. Okay? So I understand that this can be slightly confusing, but it's how we know the positions of each letters in the DNA. But there are more simple ways of doing that. Okay, so there is today these next generation sequencing instruments that help us to more easily identify the sequence of the nucleotides in the DNA. Okay, but they are an advancement from the first Sanger sequencing developed by that scientist. Okay, here the next generation sequencing involves similar steps, okay, I would say, but it allows us to read more base pairs. So the amount of data that can be processed by this next generation sequencing is much larger than compared to this Sanger sequencing. Okay, so the next generation sequencing is also known as a high throughput sequencing, and as you can imagine, it has revolutionized the field of genomics and also the molecular biology, because it allows us to sequence thousands of DNA sequences, millions of DNA molecules at the same time. So the amount of data that we can read here is much larger compared to this previous sequencing method. Okay, so here we have different sequencing technologies that work differently, but they all produce this large amount of data, okay, at a much lower cost compared to the Sanger sequencing. The next generation sequencing workflow, as you can see here, involves several steps, and I've summarized here in 4 key steps: from constructing the library you want to analyze, you want to sequence, to the actual data analysis. Okay, and let's go through these steps just so you can have like a general view of how this works. I will give you a more specific example using the Illumina, which is one that I am more familiar with because we used in our lab, but there are many types of sequencing, next generation sequencing protocols that can be used here. But the first thing is to construct your library. So this process begins with literally creating a sequencing library from the DNA sample, okay? Or sometimes from a sample that we call cDNA. I'm not going to go into details about how the cDNA is collected, but it's a type of library with the DNA that you want to sequence, okay? Then the DNA sample is fragmented into relatively shorter double-strand fragments using different methods like physical shearing or enzyme digestion. I think that's the most common, using restriction enzymes to cut the DNA and making these shorter double-strand fragments, or even using PCR amplification of specific genetic regions. Let's say that you don't want to sequence the entire DNA and you only want to sequence a certain gene, you only want to sequence a certain portion of that DNA. You can do that by only amplifying that specific genetic region, okay? Then adapter sequences specific to the sequencing technology that you want to use, such as Illumina, for instance, are then ligated to the DNA fragments, forming what we call this fragment library. Okay? So constructing the library simply means getting our DNA, breaking that DNA into smaller, shorter, double-stranded fragments, and amplifying this DNA. Okay? The second step is actually the clonal amplification. So once your library is constructed, the DNA fragments that need to be attached to a solid surface, and I'm going to show you an example in the next slide, but a solid surface, and we need to clonally amplify this DNA to increase the signal for detection during sequencing. So let's say that you have these short double-stranded fragments. We then attach those fragments to a surface, and we amplify, we make clones of these short fragments, okay? Because we need to amplify our signal to be detected during sequencing. So this process of clonal amplification actually involves binding each unique molecule of DNA in the library to beads or to a flow cell, and using PCR to amplify these fragments to create a cluster of identical clones. Okay? This step is very fundamental because it ensures that sufficient copies of the DNA fragment is there to be sequenced. Okay? We need to make sure that we have enough of those DNA fragments so we can detect them. Then the next step is actually sequencing the library. So this amplified DNA library is then subjected to a sequencing, to a specific instrument that will identify the nucleotide bases in that DNA sequence. Okay? As you can imagine, different next generation sequencing methodologies will use different ways of sequencing the DNA. Okay? They will have different ways of actually identify each individual base. Okay? But in general, this process involves cycles of DNA base synthesis, detection of those incorporated bases, and removal of products to initiate a new cycle. Okay, I'm gonna show you in the next figure, and I think it's gonna be a little bit more easier to understand what I am saying. The next step is actually analyzing the data. Okay, so the primary analysis process of this data involves getting a raw data essentially from this detector, and then we transform this raw data from this detector in these sequencing read files essentially that we call FASTQ files. Okay, and the FASTQ files are actually giant amount of sequences of DNAs that are organized by a computer essentially. Okay, then the secondary analysis that we perform in this data includes read filtering and trimming the data essentially based on the quality, based on the alignment of the reads to a reference genome, or assembly of reads for novel genomes, and many other processes. So there's like a whole thing that can go into how we analyze the data here, which is way beyond of what this course proposes. Okay, so generally this is performed by a bioinformatician a biostatistician or bioinformatician, okay, bioinformatician that will actually process these files, okay, because they are giant and they are very hard to work with. But in general here, I am showing you how this process happens with the Illumina, which is one of the most used ways or methods to sequence the DNA. So Illumina is actually a very high popular next generation sequencing technology that has been widely used for DNA sequencing applications, okay? This sequencing method is based on the use of dye terminators to determine the nucleotide sequence of the DNA molecules, all right? In the Illumina sequencing, the DNA molecules are first attached to primers on a slide and undergo a process that's called bridge amplification, okay? So here in the first process, you can see that I am showing you the live preparation. So here we have the DNA of interest, and we fragment this DNA of interest in different, shorter, double-stranded fragments. And to each of these fragments, we're gonna add adapters. Those adapters help these shorter fragments of DNAs to be placed onto these platforms, okay? So these sequences, they are then placed onto these platforms, and the adapters are specific for each methodology that you are using. So Illumina uses specific adapters, okay, for their platform right here. Once you attach your sequence of DNA, what you want to do now is actually amplify the amount of DNA, okay? So you go through several cycles of amplifications, and then you have these amplified clusters. As you can see here, each cluster was multiplied, was replicated, so you have enough DNA that can be read, that can be identified by our sequencing methodology, okay? So here we have them in clusters. So multiple copies of the same DNA fragment is localized in these clusters in a small area onto your slide, okay? So in this Illumina sequencing, we are actually extending the DNA sequence by adding one nucleotide at a time. And each nucleotide that is added at that giant sequence has a fluorescent dye. And every time you add a nucleotide with a fluorescent dye onto that sequence, there is a camera that takes an image of the nucleotide, that literally takes a picture of the nucleotide during each sequencing cycle. As you can imagine, you're adding thousands. You are doing this, you are repeating this process thousands of times, okay? And you have thousands of clusters, essentially. So your computer is detecting every single nucleotide thousands of times in thousands of sequence, in thousands of cycles, sorry. So as you can imagine, the amount of data being produced here is giant, okay? But after each imaging step, the fluorescent dye, along with a terminal 3-line blocker, they are chemically removed from DNA, which then allows for a next sequencing cycle to start, where a new nucleotide is added and then imaged, Okay, so here is kind of similar to the Sanger sequencing, because here every time we add a nucleotide with a fluorescent dye, we take a picture. But then we remove that dye, we remove that nucleotide and add another one. And then we take another picture. We remove the dye, then add another one, take a picture. And then that cycle repeats over and over. Okay, so this iterative process is actually repeated multiple times to obtain the complete DNA sequence. The images that we obtain from this process, from these fluorescently labeled nucleotides, are analyzed by a computer, and that computer determines the sequence of the DNA fragments. Essentially, that's how the computer kind of organizes it. The intensity of the fluorescent signal at each position indicates the incorporated nucleotide, and the sequence is determined based on the order of the nucleotide addition. Okay? So at each position, you are adding a nucleotide with a fluorescent dye, and then you are taking a picture. Okay? And that indicates where the nucleotide was entered. All right? And then that sequence will be determined based on the order that those nucleotides were added. Okay? And then the last step is actually analyzing the data. And analyzing the data will lead you finally to assemble the entire DNA sequence. Okay? And then you will actually know what are the letters or the nucleotide bases that make up that sequence. Okay, by knowing that, you can actually know what are the genes present in that DNA sequence that you just sequenced. Okay, how do you do that? You do that by using bioinformatics. So using bioinformatics, you can create this DNA sequence alignment, where similar sequences of bases are lined up for comparison. And this alignment will identify overlapping sequences. And with that, we can actually reconstruct the order of those sequences in the entire chromosome. By doing that, by doing this alignment analysis, we can actually, for instance, compare DNA sequences. We can also identify the genes in that DNA sequence. Okay, let's say that I have a huge DNA sequence, and I know that for the hair color, I have this type of DNA sequence here. I can align that DNA sequence for the hair color onto my DNA that I just analyzed to identify whether that whole DNA has or not the gene I was looking for. Okay? We can also use this alignment process, this bioinformatics, to find gene regulatory regions, to identify telomeric sequences, to predict, for instance, amino acid sequence, and also, this is very important, to deduce the evolutionary relationship between genes. And this is a whole new area, okay? How we use the sequence of genes to tell the evolutionary trends of those species, okay? Because in the past, to tell the evolutionary story of a species, we would use morphological data. But now we are able to actually collect DNA from those old samples and analyze that DNA sample and reconstruct the evolutionary story of that species based on the relationship of certain genes, of certain conserved genes, okay? There are specific genes that scientists look for when they are trying to understand the evolutionary trends, which is not the focus of this class, but just so you know that we can use the sequencing to actually understand the evolutionary trends, okay? So this is essentially how it works, right? So you have a sequence that you just collected, and then you have a sequence already known in a huge database, okay, that's maintained by the gene bank, and you can align the sequences and see and check whether those sequences are compatible or not. So you can tell whether those genes are the same, those genes are different, or even the sequence of the entire DNA in a particular chromosome, okay? This data is actually maintained by the National Center for Biotechnology Information, or NCBI. The NCBI compiles the gene bank. The gene bank is the largest publicly available database of DNA sequences. If you go there right now, if you type gene bank, you can literally find the sequence for millions of genes that have been already sequenced for many species, especially species that scientists use in the lab, such as mice, rats, humans, C. elegans, and Drosophila. There are tons of genes already sequenced, and you can find those genes in these in this sort of library, okay, that compiles the DNA sequences of those organisms. And honestly, if you are a public institution and if you sequence the DNA of the species that you are working, you are required to deposit those sequences in the gene bank. And every time you deposit those sequences in the gene bank, you actually get a accession number, which is like a ID number for that sequence that you analyze, for that sequence that you identified, okay? So every time that someone wants to compare the sequence that you identified, they can use this ID number to retrieve your sequence and compare to the ones that they also are studying, okay? So if you go there right now, you can find literally genes for sequences of genes for tons and tons of proteins, tons and tons of things. And with that, for instance, scientists can, create mutants by knowing that the sequence for a certain molecule in the cell, I can delete that sequence and create a mutant without that sequence, okay? And study what are the effects of the deletion of that gene in the organism. So there is a whole area here, and as you can imagine, I am talking in very general terms because there's lots of things that we could be discussing about the applications of this bioinformatics. But generally, I just want you to understand the principles of how sequencing the gene works, okay? Because that's the focus of this class, how we can sequence, how we can understand the sequence of letters of nucleotide bases in our DNA. The word BLAST stands for Basic Local Alignment Search. BLAST is a software application that we use to compare newly sequenced genomic DNA to non-sequence in the database. Remember that I told you a few slides ago that whenever you sequence a gene, whenever you know the nitrogenous bases that make up for that gene, you usually have to deposit that information in a database or in a gene bank, right? And I talked about that here. Whenever you do that, people can use this BLAST application, this software, to match their sequenced genomic DNA to that database that you are offering. Okay, so let's look at one example here. So here we are working with a gene from rats, and that's gonna be identified by Curie. Oops. So here we are working with the genes from the rat, and then here are the genes from a mice. Okay, here when we align both sequences of those two genes, one from the rats and one from the mouse, we see that there is I think here it says that there is about 93% of identity, meaning that those two sequences that we are looking at, that we are aligning, are 93% identical. They are very, very similar. Here, every time that you see one letter with a vertical line, you see that those two letters match. However, if you see one letter without that vertical line, then that means the base does not match in that location. We are looking here for the similarities between a known sequence in our mice and an unknown sequence in the rat. The known sequence that we know here is for the insulin gene. Okay, so we know in mouse that the gene should have this sequence here. Okay, now we just sequenced a genome of a rat, knowing that I have this database that this sequence for the mouse is for the insulin receptor. Having the newly sequenced DNA from the rat, I can align those two sequences and analyze whether they match with each other. In this case here, we can see that the sequence from the rat, which is shown above here, matches in 93% with 93% of identity to the sequence of the mice, meaning that probably that gene that I just sequenced might also be the gene for the insulin receptor. Why is that the case? Because besides of them, those two sequences being very similar to each other, we also know that those two species are related. Okay, so we can confidently say that the sequence in our in the species that we are studying, which is the rat, is probably the sequence for the insulin receptor as well. With that knowledge, we can identify two types of genes. We have the homologous genes and the orthologous genes. Homologous genes are genes that are evolutionary related. So here, when we do those alignments, we are able to identify those genes that are homologous. Orthologous genes are genes that are from different species, but they share a common ancestor. And here's an example. So here we are looking at the gene for the leptin, or for LEP. Okay? We can see that in a human, I am representing here with the blue letters. And for the mouse, the gene I am representing with the red letters. You can see that whenever you have a matching here, you have this vertical line. And whenever you do not have the vertical line, it's because you do not have a identity. You don't have a matching pair over there. Okay? You can see that the two genes The LAP gene in humans and the LAP gene in mouse are very similar to each other. They share very similar base pairs with each other. And because of that, we can conclude that those two sequences are orthologous genes, meaning that even though they are from different species, they probably share a common ancestor, okay? Because the two genes are very similar. The sequence of those two genes are very similar, all right? So a few slides ago, I mentioned about a Human Genome Project. This Human Genome Project was a big consortium implemented among various scientists that they decided to sequence the entire human genome to understand what are the base pairs that make up the entire, what are the nucleotides that make up for the entire DNA in the human chromosomes, okay, in all those 23 pairs of chromosomes. And we're gonna talk about some of those major findings from this human genome. But just so you know, this was a huge project, a huge effort to actually identify all the genes in the human genome. I do want to list some of the things that I found very interesting from this project, and one of them is listed right at the top, that the human genome contains over 3 billion nucleotides. That is a lot of base pairs. That is a lot of nucleotides. However, only 2% of that entire genome actually makes up for a protein coding sequence. So can you imagine that all of those nucleotides that we have, which is 3.1 billion, over 3.1 billion probably, only 2% of that actually makes up proteins in our body. The rest is all introns. The rest is everything that we do not use or don't have a specific function. Okay, so there is a lot of things that we still don't know what they are doing there and what's their function in the entire genome. Another data for me that's very impressive, and we are going to talk a little bit more about it, is that the genome sequence among different nationalities is about 99.9% similar. Can you believe that? That, you know, Americans and, for instance, Asians or South Americans or, you know, all these nationalities, we share almost of all those 3 billion nucleotides. Almost of all those 3 billion nucleotides are similar among all those nationalities. Those differences that we observe are due to these SNP and CNV. We're going to talk about this in the following slides, but can you imagine that only less than 1% of our genome makes up for the variety of human beings that we have in Earth? That for me is just like a whoa. It's very surprising to find that we actually can create so much diversity with very little of our DNA. Okay? But definitely we have a lot of things in our DNA that was already sequenced, and but very little actually makes up for protein. So this project was a project that began in 1990 under the direction of James Watson, who was one of the guys who actually discovered or proposed the structure of the DNA, right? And then they led this huge project coordinated by the Department of Energy and National Center of Human Genome Research here in the US actually. And they had this plan to sequence all the genes in the human genome, and the idea was that we could actually map the entire genome of every single of the humans, right? And the initial budget for that project was about $3 billion that was invested, and they wanted to do that within this 15 years of execution. Okay, and of course it took a really long time because sequencing that genome it takes a really long time. When you send a sample to sequence, it does definitely take a long time to you know, get the results back. It takes a lot of time to actually cut the DNA fragments in the correct size, and then prepare your sample. And then when you send it to sequencing, and then you have to analyze all that. So it definitely takes a really long time to work with that amount of data. But here, what's important in this Human Genome Project is that when this discussion started, people also started to think about the ethical and social implications of actually sequencing the genes in the human genome, because the concern here was that we would ended up creating a database where the genetic information of people would be available for everyone to access. Or for instance, you would be able to identify disease in your DNA, and do you want to know whether you have or not that disease, or do you want to know if you're gonna pass those disease to your kids or to the offspring. So there is a lot of discussion that started to arise from this project, and very important discussions, because we are working with the material genetic of people, right? And also one of the things that was raised when this project was going on is that try to find some sort of a fingerprint for criminals, right? So the idea was that you would find a criminal type of DNA, and then you would submit lots of people to this screening, so you would have those criminals kind of like removed from the society as well. So there was a lot of ethical concerns that was raised when we started dealing with personal genetic information, right? But anyways, the major features of these human genome projects. So I selected some of them, but you can definitely go back in this table and read each one of them, okay? Because they are all impressive to me, but I selected just some of them. But here, like I said, one of them was that there are less than 2% of the genome that actually codes for protein. So most of our genome does not lead to any form of protein. And you can think that you would probably think, and for many, many years, scientists thought that there was some sort of a trash DNA that wouldn't actually have any function, because if it's not coding for protein, then what is that doing there? But with the discovery of the transposon elements, for instance, that DNA is not garbage, it's not trash, it's actually, participating in generating the variability that we observe in the generations, or in the nationalities, for instance. Okay, so for many, many years, we thought that those 98% of the genome was a garbage DNA or a trash DNA, but actually, with the discovery of transposons, and as the area of genetics advanced, we actually came to realize that even though they are not coding for protein, they do serve some sort of function in the overall aspect of the human population, okay? Another finding was that probably we only have about 20,000 protein coding genes. So you can think of the innumerous proteins that we produce in our body, right? However, those various proteins that we produce in our body are coming from these 20,000 protein coding genes. So this was very kind of unexpected for scientists, right? Because we had an idea that the DNA was huge, and we thought that every single portion of the DNA was really, really important, and every single portion of the DNA would actually lead to a protein. But the number of the genes that we ended up found was much lower than the number of the predicted proteins, was even lower than the number of what we expected, right? We expected to find much more protein coding genes, but we ended up finding very little compared to the number of proteins that we actually produce. And how do we generate then those other proteins if we only have 20,000 protein coding genes? Well, probably, like I said, that's coming from small mutations, that's coming from transposon elements inserted in the DNA, or that's coming from, for instance, recombination of the chromosomes. So there is a lot of other mechanisms that generate variability, right? And we've talked about them, like the crossing over, right? So all those other factors are contributing to generate all this variety of proteins manifested in the phenotype that we can observe. Okay, so for instance, even though that the number of genes it's much lower than the number of the predicted proteins, we know that, for instance, these multiple proteins come from alternative splicing of the DNA. So alternative splicing is one of the ways, one of the means that the cells use to produce that diversity of proteins. Alternative splicing is literally what the name means, is when the DNA suffers cuts in different areas, okay? And that produces different genes, or that will lead to a production of a different mRNA, okay? Which then leads to a production of a different protein, okay? So overall, over 50% of all those genes undergo alternative splicing. And that's how one single gene can produce multiple transcripts, therefore multiple proteins. Okay, so of all those 20,000 genes that we were able to identify, most of them go through this alternative splicing, which changes the sequence, right? Which will change then the triplet codon for that specific amino acid. So with that, we are able to produce various types of proteins. Okay? Another major thing that, scientists found was that the human genomic sequence is 99.9% the same within all the nationalities, which is incredible to think, right? Because if you look at every single person in this earth, you will see that we are very different from each other, right? Even within the same nationality, we are very different. So where is that variety? Where is that diversity coming from? Well, there are two things that happen that contribute for that diversity. One is the single nucleotide polymorphism. What is that? Single nucleotide polymorphism, as the name suggests, are single base changes in the genome. Those are that point mutations that we discussed in previous classes. Okay, so mutating one nucleotide in that sequence can contribute to the entire, can contribute to a great portion of the variability that we see in humans, okay? We also have these CNVs, which stand for copy number variations, which is when segments of DNAs are duplicated or deleted, which kind of resembles that alternative splicing, okay? But with that, in this slide, I want you to realize that this concept, and I think I discussed when I discussed about population, genetic population, is that race is a very it's a non-scientific concept, okay? Because we don't have different races, because we all share this very similar genetic information, right? And so, like I said, the idea of the terminology, involved with race came from this social Darwinism, where people, looked at people that were physically different from each other, and they thought that just because they were different, they were less of a human, essentially, okay? And that's why many bad things happened in history, okay? So the concept of race is genetically not true, okay? It's scientifically not possible, because we do share almost 100% of similarity in our genome. So, like I said, the concept of race is very associated with this social Darwinism, where one, with coding marks here, one race thought that was bigger than the other or better than the other. So this was one of the things that the Human Genome Project also contributed to kind of like demystify, okay? So the other thing that came from this contribution is that, with the Human Genome Project, we were able to identify many genes involved with genetic disease, and with that we were also able to develop new treatment strategies to target those diseases, because now we know that those diseases, they have a genetic background, and I know what are the genes that they are affecting. Therefore, I can create more specific therapies to target those genes that perhaps are not working as properly, not producing the right protein, okay? So with that, we were able to create these genetic maps, okay, where we are able to actually identify the genetic disease or the predisposition of genetic disease in humans. So as you can think, there was a lot of good things, and also lots of concerns that came from this initiative. I promise you that we are heading towards the end of our lecture. I think I have probably 10 more slides to go, and I am pretty sure that this is gonna fly and you will not even realize that. But in these final slides, I just want to talk about the transcriptome and proteomics, okay? So far we've been talking about the genomics and how we work with the DNA, how we sequence the DNA, why is it important to sequence the DNA, what are the advancements that we obtained from sequencing the DNA. But one thing that we can do is also looking at the transcriptome. We can look at the transcriptome. The transcriptome, if you remember the central dogma, you have the DNA, which is transcribed in RNA, which is then translated to protein. Okay? And please remember this as if your life depended on this, because it does. All right? So from now on, as a biologist, you should not forget about the central dogma. So we were looking at the genomics, we were looking at the DNA. So transcriptome actually looks at the RNA. And when we look at the RNA, we are actually looking at that portion of the DNA that actually will become a protein. So that approach can actually be a little bit more, let's say, the genome is, giving the whole information about the genetic profile of that organism. But when you are doing the transcriptome, you are looking at the specific part of that genome that gives you a protein. Okay, so here the transcriptome analysis will reveal a profile of the genes that are expressed in the cells and tissues of that organism. Only the ones that are expressed, okay, because they will become RNA, and then from RNA they will become protein. So transcriptome analysis, or also known as transcriptome, is actually a global analysis of gene expression. Okay, so those can be that this global analysis can be qualitatively or quantitatively. Here, qualitatively, we can actually identify which genes are expressed and which genes are not expressed. But quantitatively, I can also know how much of those genes are being expressed. So I can quantify the transcripts. I can quantify the amount of mRNA produced by that organism. Okay, not only I can identify which mRNAs are produced or are expressed or not, but I can also quantify. I can measure the levels of expression of those different genes. How do we do that? I'm going to talk about two specific techniques here, but you don't have to go much into details, okay? There is definitely one video here in my slides, and you can look over those videos of that video, but don't get too attached into details about this, okay? Because like I said, there is a lot of things that can be done different depending on the labs, depending on the type of technique that you use. So there is a whole new world here about this. So the first thing, one of the first techniques is the DNA microarray analysis. And I would like to jump to my schematic figure to explain how this works. So first, in this DNA microanalysis, we isolate the mRNA. So we get the tissue sample from the organism and we separate the mRNA molecules. The way scientists separate the mRNA molecules are various, okay? So there are many ways that you can collect only the mRNA molecules. From the mRNA molecules, you make cDNA. cDNA is complementary DNA. And here we make cDNA by using the reverse transcriptase, which is an enzyme that will convert RNA and DNA. Okay, so the reverse transcriptase will make the transcription process in the opposite way. Okay, so from the RNA we'll make DNA. So here we make cDNA. And here, making the cDNA, we are gonna use fluorescently labeled nucleotides. So those nucleotides that we are making this new DNA are or can be identified via fluorescent techniques. Okay, so at the end of this process we have labeled molecules of cDNA, okay, as indicated here. Then the next step is the hybridization. So we are gonna put that cDNA library in a DNA microarray. The microarray, it's literally a slide, okay, a very tiny slide. And those slides, they have little wells. And each spot of that slide, of that microarray, there are millions of copies of short single-stranded DNA molecules for different genes, okay? So here, for instance, you are, let's say, looking for a specific gene. You want to know if your organism expressed or not the gene for insulin. Okay, let's go back to the example of the rat and the mouse. You want to know whether your rat expressed the gene for the insulin receptor. So here, your microarray will be loaded with DNA, with short single-stranded DNA containing the gene of interest. Okay, and if in your sample you have that gene, this complementation will happen, and this complementation we call hybridization. So your cDNA will hybridize to the DNA onto the microarray. Okay, after that happens, you put that microarray in a scanner, and the scanner will measure the fluorescent signal of each spot, measuring and when it shoots the fluorescent light onto your samples, it can measure the amount of mRNA expressed in each spot. And that tells you how much of that gene is being expressed in your tissue sample. So for instance, when you have no fluorescence, that means that no gene was expressed in the gene sample. If you have, for instance, a moderate fluorescence, that means low gene expression. But if you have a bright signal, you have highly expressed gene in that tissue sample. Okay? But there is a way of being more specific, because here we are just comparing the intensity of the light. We're not actually getting a number of how many mRNAs we have in that sample. Okay? There is a way, and that way is called RNA sequencing, and that's what I'm going to talk about in the next slide, to know how much of mRNA there is in that sample. Okay? If you have more questions about the DNA microarrays work, I'd recommend you watching this video, because it goes very step by step in this process. Okay? But we have this RNA sequencing, or RNA-seq. This methodology is also known as a whole transcriptome shotgun sequencing, as if you collect all the mRNAs in that sample and you sequence every single one of those mRNAs. Here you actually have a quantitative analysis of those RNAs expressed in the tissues. Okay? This also provides you actual sequence data, can be carried out in situ, and this is a very common technique used in labs nowadays to actually identify the genes of interest, or to actually identify the genes present in your organism, in the organism of interest, in the organism that you are studying. Here is a very summarized workflow of how the RNA sequencing works. So the first step is to isolate the RNA from the samples. So here we start by extracting the RNA from the cells. The RNA is the molecule that carries the genetic instruction for what? Building the protein. So it is essential to isolate the RNA to understand what are the genes that are active in that sample. What are the genes that will produce a protein in the sample? The second step is making fragments of RNAs. So you get the RNA and fragment it into short segments. So once you have the RNA is a very long and fragile molecule. So we can't actually sequence that molecule directly. So we ended up breaking that long RNA into smaller and more manageable fragments, and we prepare those fragments for sequencing. The next step is to convert the RNA fragments into cDNA. So here, when we talked about the next generation sequencing methodology, like the Illumina, we focused on how that equipment works with DNA. Okay, so here what we need to do is to convert that RNA fragment into its original DNA. And we do that by using the reverse transcriptase, okay, that does the opposite process, that uses the RNA molecule and produces a DNA molecule. Then the fourth step here is to connect that cDNA to adapters and then amplify those cDNAs. Here the process is just like the sequencing that we talked at the beginning of this class. But very briefly, what's happening here? So at this step, we insert these short adapter sequences to both ends of each cDNA fragment. As you can see here, we added these adapters at the end of the cDNA. They are essentials. These adapters are important because they allow the fragments of cDNA to attach to that flow cell, to that platform that we use to sequence, right? That platform, which is the surface where the sequencing will occur. These adapters, they contain this sort of a barcode that helps afterwards identify your sample if you are actually using multiple samples in the same sequencing run. Okay, this is just an extra information. You probably will work with that if you go to a molecular biology lab. Okay, then at this portion we also need to amplify the signal, right? Remember that the amplification here works with that bridge formation, right? So you have clusters of that cDNA in your flow cell, for instance, and then in those clusters you amplify the amount of DNA in each cluster to sort of amplify your signal. Okay, so amplification is important, and we are using PCR here to ensure that we have enough DNA for a correct sequencing. Okay, let's just think like you're making copies of a library book, right? You need multiple copies to ensure that there is enough book or there is enough copies for everyone to read. Okay, so similar here, we amplify these fragments to make the sequencing processing more efficient and more reliable as well. Then the other step is to perform the next generation sequencing. The next generation sequencing, as we already talked, is a high throughput sequencing method that can read millions of DNA fragments at the same time. And that will generate a very huge amount of data. Okay? In this step, the cDNA fragments with adapters onto that platform are loaded into the next generation sequencing machine, like the Illumina machine that I presented to you before. So inside the machine, the fragments, they bind to that platform and they form that bridge amplification where the clusters of identical fragments on the cell surface, they are amplified, right? The sequencer, like I said, will read the DNA bases A, T, C, G one by one by adding fluorescently labeled nucleotides. So as you incorporate each base when you are amplifying, each base is carrying a signal. So each base will emit a specific color of light. And your machine, your Illumina machine, for instance, will detect and record that. And it knows that if it detects red, that's adenosine, or if it detects green, that's a cytosine, for instance. Okay, I'm just giving you an example. Doesn't mean that that's what's happening with the Illumina. So the output of this sequencer is actually a very large file that contains the reads. Reads are the short sequences of the nucleotides that represent the cDNA fragments. Okay, so these reads are the building blocks that we want to actually understand what are the genes that are present or active in the original RNA sample. The final step is to analyze the data, and then by analyzing the data, we mean we are aligning those sequences that we got with a reference genome or a reference transcriptome, okay, to understand whether those genes are present in the sample that we collected. Okay, it's a very more complicated process than 6 steps that I presented here, but it's a very important and interesting process. Okay, the last 2 slides, and I promise we're going towards the end of the lecture, I just want to talk about proteomics. Proteomics, now go back to our central dogma, okay? So proteomics will now deal with the presence of protein, is the analysis of the amount and composition of the proteins in those cells. So proteomics is the identification, characterization, and quantitative analysis of all the proteins expressed in the cell of your organism of study. And the complete set of all those proteins that are present in that sample is known as proteomes. The same thing that we had the transcriptomes and the genome, now we have the proteomes, okay, which is the group of all proteins encoded by the genome. Here, with this technique, by knowing the proteins expressed in that tissue, we can, for instance, compare a normal tissue with a diseased tissue and know whether those proteins are abnormal, whether those proteins are functional or not. And with that, you can actually create biomarkers for disease conditions. Okay, so if you know what a normal protein should look like, you can identify a protein when it's abnormal. Okay, so proteomics, and this is my last slide. Proteomics is the area of science that will provide you information about protein structure and function. We'll also tell you about post-translational modifications, because the mRNA, even though the mRNA will become a protein, that mRNA can undergo changes, and then that protein can also undergo changes, okay, after it has been translated. Proteomics can also give you information about protein-protein interaction, protein and metabolite interactions, okay? We can also know the cellular location of proteins by labeling those proteins in the cells. We can also, for instance, study the evolutionary history of certain proteins, for instance, like the example I gave to you, the presence of the insulin receptor, right? If that came or not from the common ancestor, if more than one species share that same protein or not. So we can also use the protein profiling to tell the evolutive history of that species. Okay, so to finish this class, and I know I've talked about a lot of interesting information, I just wanted to give you a chance of knowing that, genetics doesn't have to be all about Mendel, okay? It doesn't have to be all about Punnett square, or, it can be more interesting than that. So In this course, we actually started from, working with A's, B's, genes to actually understand how we can identify what those genes are. So I hope you enjoyed this class, and you can see more what we do nowadays with all this knowledge that Mendel and Morgan one day provided us.